Well, thank you very much, Dominic.
Yes.
I hope you could take the chance and have a coffee in the meantime.
Not
And
really.
before we are discussing now the last session of today on these presentations, I'm going to tell about some other trends in novel foods we are currently facing, which are plant extracts, cannabidiol, and also some novel foods with engineered nanomaterials, which I will hand over then to Anne-Maria and Andrea Termini, my colleagues from the nutrition unit. But before, I would like to express one issue, because all these presentations have been conducted by the Nutri unit, and in particular by the novel food team. I would like to stress that all these presentations, all the information we have there presented, are based on the novel food guidance, which was developed and adopted by our panel, and also based on a number of previous assessments of our experts in the novel food working group and in the panel. So all the output what we are presenting here is rather observations of how our panel is approaching such novel foods. We didn't want to put additional work on our panel, so that's why this preparation and presentation comes from the unit itself. But actually, the way how such novel foods are assessed is from the panel, of course. Yeah. So these are the three main topics I would like to present in the following slides together with my colleagues. And I would also like to express some particularities with regards to food supplements, and especially, of course, novel foods in food supplements. You may be aware that we have a directive on food supplements from 2000, and a directive as such is a legislation that is giving a lot of leeway to member states with regards to the implementation and how they are actually implementing these requirements on their level of member states. And it's different from regulations, of course, where the Commission is drafting implementing rules and where we have directly implementing rules centralized, applicable for all of Europe. So in this food supplement directive from 2000, we have a list of vitamins and minerals in Annex 1 and 2. And EFSA did quite a lot of work on this annex as such, because in Article 4, it was expressed that EFSA basically should assess all the vitamins and minerals on the market at that time. And as such, as a consequence, EFSA assessed almost 300 of vitamins and minerals. Basically, that was done largely by the food additive panel. Directive, as I said before, we don't have...
Excuse me, Wolfgang.
Yeah.
The slide.
Yes. What is the slide?
I still the question and answer slide, or is it my problem? Excuse me, Wolfgang.
No problem. So there is no harmonized, centralized approach how to implement this legislation, but it is on member state level. There is also no harmonized maximum or minimum level for vitamins and minerals, what can be used in food supplements, and also with regards to other substances, and that includes a big number of botanicals, where there are no harmonized EU-wide assessments done. Nevertheless, EFSA has still a big role in food supplements in the EU, because this food supplement directive is also expressing that when setting maximum or minimum levels for vitamins and minerals, upper levels and dietary reference values should be considered when imposing national limits. And there, of course, that's the task of the NDA panel, to come up with or to establish upper levels and dietary reference values. EFSA has also another role whenever risk managers or also on our own initiative, EFSA could self-task work and do some safety assessments on substances, either those in the annex of this directive or on other substances. And of course, when it comes to novel foods, which are intended for food supplements, then we have, of course, the novel food regulation, and then also it's the NDA panel, which is assessing the safety of novel foods, also when they are for food supplements intended. So I've mentioned already before, the role of our panel is to set up DRVs and including upper levels for vitamins and minerals. And we also EFSA is also could be tasked with regards to the safety assessment of other substances. Then we also have a working group on health claims, which is an efficacy assessment. But also there, or let's say there, EFSA has been asked to hold the assessment on botanicals. And so the task of our panel is, in addition to the above, at the moment largely nutrient sources and novel foods when they are intended for the use in food supplements. And of course, other EFSA panel could be also involved in some issues related to food supplements when it comes to food colors or GMOs. I would like now to highlight some specificities with regards to food supplements that are not necessarily, or they are not applicable with other foods, or to a less extent in other novel foods. And one is the target population. Food supplements is among the very few food categories where applicants could limit their safety data to very specific target populations, such as adults, for example. And then also EFSA would restrict its assessment only for such food supplements when they are intended for adults to this target population. Such a restriction is not applicable when a novel food is intended to be added to other foods, such as breakfast cereals, beverages, or dairy products, breads, and so on, simply because risk managers consider that such food products could not be restricted to be consumed by only very few limited target population, but basically the whole general population could consume such products. And therefore, whenever a novel food is intended for such food categories other than food supplements, other than infant formula, other than foods for special medical purposes, then usually we have to assess the safety for the general population. If an applicant wants to limit the target population for his novel food, for food supplements, then the applicant should also give some reasoning why he wants to restrict it. And the reason is not because it's not allowed, but whenever such a restriction is proposed by applicants, then our experts and the panel is asking, yeah, but why? Are there any data? Are there any reasons why such a food should not be consumed by pregnant women? And then, of course, they would like to know what are the reasoning behind, and whether these reasonings, some concerns, may also be applicable to other population groups. So that reasoning should always be given. What is easier for food supplements is the exposure assessment, because that could be limited basically to the maximum proposed intake levels. Sometimes also background intake should be taken into account, especially when it comes to vitamins, minerals, other micronutrients. Then the exposure assessment can be limited to the maximum intake and taking into account also background. Then there are also some specificities with regards to the history of use of food supplements. These are maybe more regulatory considerations, but you may be interested in, is food supplements when they have a history of use in food supplements only in other countries. Then such history of use would not qualify to argue that such a novel food has a history, such a traditional novel food has a history in another country, and it would not qualify for such traditional foods, which are very specific groups of novel foods. History of use in food supplements within the EU, also there, when there is only a history of use in food supplements in the EU before 1997, whenever an applicant would like to make use of such an ingredient in food supplements and he wants to add it in other type of foods, then you would need to make an application, and then the history of use food supplement does not would not as such qualify that you can use it also in all other kind of foods. And then also interesting is the history of use as a drug, because we are facing let's say some ingredients that have a history of a use as a drug in other countries or maybe even within the EU. And they would really like to highlight that the different approach our panel is applying for novel foods than the approach what is normally used for drugs, which is of course in drugs we do the drug authorities, they do a risk-benefit assessment. To some extent, side effects may be acceptable. Usually you have a very specific target population group. You use drugs normally very limited time, or in some cases maybe also long time, but often it's only intended for short-term use. And of course, such information on the history of a drug can provide important information with regards to the safety of an ingredient of a novel food. But it is very difficult or if not impossible to establish the safety of a food on the basis of a history as a drug. In general, because today we have talked very little about the history of use, in general our panel gives rather limited weight on a history of use of a novel food in other countries. And the reason is, first of all, reporting. And especially when it comes to complex novel foods, especially like those plant extracts, where there is a big diversity of the source of the raw material, from the processing, from historical conditions of use, our panel gives normally little weight to such data. It's very difficult, if not impossible, to establish the safety of a novel food on entirely history of use data. Nevertheless, such data should be provided because they really could provide important information with regards to the safety of an item. I'm coming now to very few other aspects on plant extracts, particularly some aspects that have been maybe discussed less in the other areas. And this is a picture of some previous plants that have been used to extract some components out of it and which were subject of a novel food application and which have been assessed or which are under assessment by our panel. And I would like to illustrate the diversity alone from the source. We had assessed an polyphenol-rich extract from cranberry juice. So the cranberry juice is highly consumed in some countries. And one company tried to extract the proanthocyanidins fraction out of this juice that is consumed. Yeah? So there the juice had quite, the history of use of the juice had a quite big weight in the assessment, and our panel took such history of use of the juice into account, in particular also because the quality and the quantity of the proanthocyanidins was very similar or not more than what is consumed from the juice as such. And then we have plants that have been used for thousands of years, such as the olive tree for oil production. But normally people are consuming the oil or the olives, but not the olive leaves. And we had a few years ago assessed an extract, hydroxytyrosine, from such olive leaves. And of course, the leaves may have a different composition from the fruits, and that has implications for the risk assessment. We're also facing some applications based on plants from Asia, in particularly, not only, but maybe more than from other continents, which have a history of use in Chinese medicine. On the right side, that's Cistantia tubulosa, and a parasitic plant that is currently assessed, which is under assessment. Actually, it was adopted recently. We have here on the very left side some fruits from Southeast Asia that have a history of consumption there, but not in Europe. So here we have very little information about such plants. Curcuma extract, of course, we consume the curcuma, but when you extract some fraction out of it, the safety of such an extract may be very different from the original source. And very recently, we have received an application that's again very different from all the others, which is an apple cell extract, from cells that have been cultured in bioreactors. And of course, all these different sources have big implications on how our panel is giving weight and what kind of data are requested for the various different applications. And to make things even more diverse and complex, it's not only the source, which is very diverse, but also the production process, of course, whether the whole plant is used, fruits, the leaves, the roots, the stems. Then we have varieties in the production process, different extraction solvents used. It's very big difference whether you're using water, hot water, or ethanol. Fractionizing, enrichment, concentration steps. So all these are further multiplying the diversity of such products. Very often, or most of the cases, we are facing mixtures of plant mixtures from either more plants, but also within one extract from one plant. Usually, we are dealing with mixtures composed by hundreds of thousands or more components. One of our main focus when approaching such applications is the hazard identification, because usually we are dealing with mixtures. Often we are dealing with sources where we have very little knowledge in Europe, with no history of use as a food nor as a drug or whatever. So what we have established in EFSA is, first of all, an EFSA compendium on botanicals below the link. That's an extraordinary work from a working group from the scientific committee on plants, which all this work has been used to establish such a database that is now currently covering more than 2,700 plants, more than 2 million scientific abstracts have been evaluated, more than 15,000 full-text articles. And this is definitely an important source that we in EFSA, but also applicants should consult when they are dealing with such applications. And in addition, Andrea has mentioned at the very beginning, with the new regulation now, EFSA has the sole duty in risk assessment of novel foods. We lost a lot of competence from member states who were previously involved. So we have implemented a literature search. We started with botanicals, plant-based novel foods, but we have extended it to all other novel foods, which is done in a very good collaboration with the University of Prague. which is performing a literature search. We have discussed with them a lot on the approach. We're using all the big databases. We're using generic keywords regarding toxicology, adverse events, effects, but also more targeted keywords with regards to particular components of concerns like alkaloids, for example. Yeah. So that is clearly a key step in the risk assessment of such novel foods. For that, obviously, it's important to know something about the identity. We have stressed, colleagues before have stressed the importance of the composition of a good characterization of a novel food, and that may be very particularly important for such botanical extracts, which are usually mixtures of many different components, and where we always have at the end, nevertheless, despite all efforts, a certain fraction of unidentified, uncharacterized components. Manufacturing process, we know that is important, particularly the extraction solvent use, but also the other steps. Yes, one thing I would like to highlight here is that also the applicant should perform a literature search. He should not rely on EFSA only. And there's also an advantage for applicants is that if they find some things, some issues that may not be in favor of their dossier, they could proactively address such findings before EFSA is finding it and confronting the applicant with such information. And then some other aspects in the safety assessment of such extracts. Very often, when you use food supplements, of course, a consumer would like to know what it is good for, and that's the reason why usually that's this case, that an applicant has certain presumed beneficial effects in mind, and often there are also in the dossiers we find references, scientific articles on presumed beneficial effects. And the way our experts are approaching such information is we will not, or our experts do not look whether these beneficial effects are true or not true in the safety assessment of novel foods, but they rather look from a safety perspective on such effects. They would like to see what is the mode of action, if there is one. And beneficial effects, they may be beneficial for the one population group, but not necessarily for other population groups or for specific conditions of use. So our experts are looking from this perspective when it comes to such information on presumed beneficial effects, which is very often the case seen in such applications. And then regarding toxicological aspects, I've mentioned before, we almost always have a mixture when it comes to such plant extracts per se are usually mixtures. We can only stress how important it is to do a very good characterization and to limit the proportion of the uncharacterized fraction. But this nevertheless has some implications also for the toxicological testing approach. When it comes to toxicological tests, it's extremely important here to really have a representative testing material. And that is very often limited when it comes to published articles. There's usually limited information on compositional data. And unfortunately, we also have seen toxicological study reports, which are conducting repeated tox studies in animals, and where there was at the end extremely limited, almost no information about the test materials. And then it is very, very difficult, if not impossible, for our panel to accept such studies if it cannot be shown that the test material was really representative for the novel food in question. And because we are facing usually mixtures in such novel foods. It is also the default toxicological approach with applying a factor 200 on a subchronic study that's almost always applied by our panel, and the reasoning is very simple because of this uncharacterized fraction. Because it is whatever additional data are provided on human studies or other studies, it is very difficult to address this uncertainty that comes from the uncharacterized fraction. And that's why usually our panel is applying the 200 here on subchronic studies. And they are less willing to reduce this, particularly for this type of novel foods. And then also very complicated is the genotoxicity assessment because, again, of such mixtures. And for that, I would like to hand over to Anne-Maria to discuss about the complexity of genotoxicity assessment of such novel foods. Thank you.
Anna-Maria, we can't hear you.
Sorry, hello, I was muted. Hello to everyone, and thank you, Wolfgang, and thank you all for your participation today to this open plenary. My name is Anna-Maria Rossi, and today I'm going to give you two short presentations. One is focused on the genotoxicity assessment of novel food, and the second one on the synthetic cannabidiol. So, starting from the genotoxicity assessment, also the novel food follows the EFSA genotoxicity testing strategy as presented in the EFSA Scientific Committee opinion of 2011. Basically, the genotoxicity strategy is a stepwise approach. The tier 1 comprises a basic battery of in vitro tests that investigate the different genotoxicity mechanisms. The tests recommended are the bacterial reverse mutation assay and the in vitro mammalian cell micronucleus test. The bacterial reverse mutation assay assess the potential of the novel food to induce gene mutation, while the in vitro micronucleus assay identifies substances that cause structural and numerical chromosomal damage in cells that has undergone cell division during and after exposure to the test substance. In case of the positive results of the in vitro micronucleus test, if combined with a kinetochore staining or a fluorescent in situ hybridization, there is the additional information on whether the tested substance induces clastogenicity or aneogenicity. And this is very important since it will allow to better select the possible in vivo follow-up studies. Only if one of the in vitro tests will result positive, there will be a follow-up to investigate whether the positive results can also be observed in vivo. The criteria to select the proper in vivo studies are, of course, the genotoxicity mechanism detected in vitro. For example, if a substance appears to be clastogenic in vitro, of course, the in vivo test selected should have as an endpoint the assessment of the clastogenicity effect. One of the critical issues of the in vivo studies is that often they are limited to certain tissue restriction. Therefore, it is necessary to provide evidence of the target cells exposure. For example, if there are no evidence of bone marrow exposure, a negative in vivo micronucleus study cannot rule out the concern for genotoxicity. The EFSA Scientific Committee published in 2017 a clarification of some of these aspects. Additionally, The guidance of 2011 provide also some example of different scenario for the in vivo follow-up. The other major topics regarding the genotoxicity assessment is the genotoxicity assessment of the mixture. Indeed, from the presentation given from my colleagues today, it is evident that the novel food are offered mixture, and this is a challenge in the genotoxicity assessment. In 2018, a statement has been published by EFSA specifying how possibly proceed to the evaluation of the mixture. Clinical mixture can be defined, can be identified as chemically fully defined mixtures for which a component-based approach will be followed, or mixture containing fraction of unidentified components, in which the identified components will be assessed via component-based approach, while the unidentified fraction should be assessed separately, and if not feasible, via a whole mixture approach. Just to clarify this concept, if we have a fully chemically defined mixture for which we identify a genotoxic substance via a substance which is genotoxic via a relevant route of administration, there will be a safety concern for the genotoxicity, and therefore the substance will be considered genotoxic. If the mixture will contain one potential genotoxic component, so one substance for which genotoxicity is not fully assessed but can give rise of concern, can raise some concern, there will be the need to provide additional data according to the EFSA guidance. Of course, if there are no genotoxic substance present, then there will be no safety concern for genotoxicity. However, as in the novel food area, we deal mostly, if not all the time, with mixture for which some of the chemicals have not been characterized. In this case, we proceed as follows. We do, of course, first the characterization, the evaluation of the genotoxicity in the characterized part of the mixture. If this part of the mixture contain a genotoxic substance, then of course there will be a safety concern. If there are no genotoxic substance, then we will proceed to evaluation of the uncharacterized part of the mixture, which ideally should be separated by the characterized form and tested separately, once again according to the EFSA guidelines. Often this is not possible, and in this case, the whole mixture approach should be implemented. So basically, the entire mixture will be tested, and again, we will follow the EFSA guidance. Of course, this is what normally we do, and the evaluation of the whole mixture approach is not always so straightforward. Okay, I will now instead like to talk a little bit on the synthetic cannabidiol. So the novel food catalogs is a non-exhaustive list that serves as an orientation on whether a product will need an authorization under the novel food regulation. It is based on information provided by the EU member states, and it is managed by the Commission. According to this catalogue, cultivation of cannabis sativa is permitted provided that they are registered in the EU's common catalogues of varieties of agriculture plant species, and provided that the tetrahydrocannabidiol content does not exceed the 2%, the 0.2%. Extract of cannabis sativa are derived products containing cannabinoids and are considered as novel food. Synthetically obtained cannabinoids are considered as novel food. Presently, we have in EFSA 3 cannabidiol under assessment for under risk assessment, pending further decision from the Commission. We know that EMA has recently approved Epidiolex, which is an active substance of which the active substance is actually cannabidiol, derived from the milled botanical raw material of Cannabis sativa. Epidiolex is an adjunctive therapy for seizures associated with Lennox-Gastaut syndrome or Dravet syndrome. And the indication is for patients of 2 years of age and older. Should be underlined that the Epidiolex, that these diseases and these syndromes are actually intractable childhood epilepsy. Okay, cannabidiol is under assessment also in EFSA, and EFSA will perform a completely independent risk assessment for the following reason. That first of all, the assessment at EFSA for novel food is not based on risk-benefit. Additionally, EFSA is targeting general population and not patients. And in addition to this, we should consider that the synthetic cannabidiol under assessment are produced, of course, via a different production process. And in addition, just to clarify that, like Wolfgang said previously, that cannabidiol will follow the approach of the EFSA NDA guidance for novel food. I think I try to go through these topics very fast because I know that we are tight with the time, and I will leave the floor to Andrea. Thank you. I will be happy. Oh.
Andrea, you're mute.
Sorry again, on the other screen. Just to say that I will go to the essential element of the presentation in the interest of time, because I see we are a bit behind schedule. So therefore, I restricted these to 2 slides where I will highlight you the main concept, but I'll be happy to answer questions there in the Q&A. As we said at the beginning, the Novel Food Regulation is actually the first piece of legislation which mention nanomaterial. That happens not only because the technology is novel per se, but also because the regulation was revised at the time in which nanomaterial were exploding and appearing also potentially in food. So that's where it was regulated first. This means also that the regulation of novel food, as you see on the slide, is also the one providing the Commission definition on nanomaterial, which I won't read in the interest of time, but it has to do with size and properties of the material that are to be considered as nano. EFSA has been taking nanomaterial under scrutiny for quite some time, and the 2 essential pieces of guidances were provided by the scientific committee. Actually, one has been provided in 2018, and the second is under finalization this month, following a public consultation. The first one has to do with engineered nanomaterial, which are those products which are engineered on purpose to act as nanomaterials. So they have the size and expected properties by design to be nanomaterial, and they fall under this guidance. And for this one, we, as I mentioned at the beginning, we receive one novel food application of a product intended to be used as engineered nanomaterial, and in particular, that is proposed as a source of iron. The second piece of guidance, which is essential in this area, which has taken up quite some space in our risk assessment and in other areas of EFSA, is the evaluation of those products which may contain small particles, including nanoparticles. This is a guidance which has been, as I mentioned, under public consultation, and is under finalization right now by the scientific committee working group. This is a tricky part of the assessment because by using new methodologies, it became more and more evident that some of the processes that are used, particularly in some areas, take one of the most impactful example in our area is sources of nutrients, for instance, of reducing the size of products to increase bioavailability, for instance, in the case of sources of nutrients, results often in the presence of nanoparticles being present in the food. In that case, we do have at the moment several novel food applications which were submitted as a standard novel food application, but for which we are investigating the potential presence of nanoparticles. This was unexpected at the beginning of the assessment, but while applying the methodology described in the guidance of the scientific committee, it became evident that there was potential of nanoparticles being present, and therefore EFSA as taking up the duty of assessing those nanoparticles present in the food. Yeah, I think in the interest of time, I will stop here. These are the two main pieces of regulation that would apply here, and that the applicants are encouraged to follow. And if there are more questions, then I will take them under the Q&A session there afterwards. So I think my presentation concludes this third session, and then Dominique, please, you can open up the floor for the Q&A.
Okay.